Okay. Greetings again. Welcome to this unit called Microbiology. This is our second lesson. And this is our second lesson on this date of 19th of November. I believe we are all in the correct class, the applied biology class. So let's start off. And first of all, let's start by each member ensuring that he or she has already checked off the attendance in the LMS. That is important. It is good to confirm your attendance for this class. And then we start. Today's topic is called sterilization. Yes, maybe just to have a brief recap on our previous lesson, we had studied on the topic called safety, where we learned about the different types of labs. We also studied about the basic or the lab infections, the basic lab infections which can occur in the laboratory. Furthermore, we went through the basic safety procedures in the lab, and we completed by studying the WHO, the World Health Organization Code of Practice with regards to microbiology, where one of the important aspects we went through was the biohazard sign, which is used to indicate the safety levels in the lab. So for today, we are going to go through the topic called sterilization. Our learning outcomes for this session will be, by the end of this lesson, each trainee should be able to explain the meaning of the terms, and secondly, should explain the importance of sterilization. Lastly, should describe the methods of sterilization. To start with the definition, sterilization refers to making a substance free from all microorganisms, both in vegetative and sporing states. One important aspect about the microorganisms is that, especially the bacteria, they form spores when exposed to stressful conditions. So sterilization is supposed to clear the bacteria both in its vegetative state and also in its sporing state. Terms used in sterilization include disinfection, which refers to the destruction or removal of all pathogenic organisms capable of giving rise to infection. However, it does not affect the spore state of organisms. Antisepsis refers to the prevention of infection, usually by inhibiting the growth of bacteria in wounds or tissues by using agents called antiseptics. Decontamination is the process of rendering an article or area free of contaminants, including microbial, chemical, radioactive, and other hazards. Cleaning, which we all know is just the process of removing soil or dirt as a preparation step before employing the sterilization or disinfection procedures. Germicide, these are substances which kill bacteria. They are also known as bactericidal agents. Another term which we can include there is bacteriostatic agent. These are agents or substances which do not necessarily kill the bacteria, but they just stop or slow its growth. Asepsis is the state of being free from bacteria, viruses, and other microorganisms. Importance of sterilization. It's important, or sterilization is important in the preparation of food substances such as pasteurized milk. What is the meaning, or maybe the definition of pasteurization? I'd like someone to try give us an explanation of that term, pasteurization.
I can try.
Yes.
I would say that pasteurization is the process of eliminating the microorganic component of milk.
Thank you. Eric has said pasteurization is the process of eliminating microorganisms from milk. Is there another trial? Shamim? Or Vivian? I can say pasteurization is the process where we heat milk in order to, let's say it's not necessarily above 100 degrees Celsius, but just below, which is a temperature which is just enough to kill the microorganisms but not to destroy the food substance itself, which is the milk. It does not necessarily make the milk sterile, but it does kill most of the microorganisms. Okay, another importance is the preparation of surgical instruments for performing surgical operations on humans and animals. This prevents the spread of disease through the instruments. Another important aspect of sterilization is it prevents infections in wounds, like through use of antiseptics. It is also important in enabling the successful performance of experiments in the microbiology lab, because it removes all the other microorganisms which are not targeted and only leaves the microorganism of interest to be studied. Finally, it is important in preventing the spread of disease in humans and animals. This is by cleaning surfaces which humans come into contact with. There's a term called fomites, which includes the microorganisms which are found on surfaces. So it protects both the patient and even the professional who come into contact with surfaces. Methods of sterilization. There are two methods of sterilization, two categories of these methods of sterilization. One method is physical methods, the other is chemical methods, and they are classified based on the manner of agents used in the sterilization process. Physical methods, as we can see there, include sunlight. An example is UV. Drying, an example is desiccation. This refers to removal of moisture, which is usually important in enzyme activity. Drying or dry heat, such as incineration, is a reliable method of sterilization. Moist heat, such as pasteurization, which we've covered. Filtration such as filter membranes. An example is filtering samples in the lab, where you filter out the bacteria. Actually, you filter out the viruses. You filter out the bacteria while allowing the virus to pass through the filter membrane, and then you collect the virus for study. The last example is radiation, where we have an example of ionizing radiation, which is used to sterilize equipment. That is a picture of a moist heat sterilization equipment. That is an autoclave. One is the one which is commonly used, the other is a bit advanced. That is a medical incinerator, which is an example of dry heat sterilization method. There's a term called thermal death time. This term is used in referring to the heat methods which are used in sterilization, and it is the minimum time required to kill a suspension of microorganism at a specific temperature in a specific environment. Chemical methods include alcohols, aldehydes, halogens such as chlorine, phenols, surface active agents, also known as surfactants. These include detergents. We have metallic salts such as silver nitrate, and gases such as ethylene, formaldehyde, and BPL. These chemical agents, they act through the following methods. First, there's some or a number of them act through coagulating the protein found on the microorganism, thereby destroying its integrity. The second method is disruption of cell membrane, resulting in exposure, damage, or loss of the cell contents. The third method of action is removal of the sulfhydryl group, which is essential for the normal functioning of enzymes, the enzymes of the microorganism. So once these enzymes have been deactivated, the microorganism will be destroyed. And lastly, through substrate competition, where both the chemical agent and the microorganisms compete for the substrate, and thereby one will be eliminated. We've come to the end of the class, and we've seen all the methods which are used in sterilization. We've covered the importance of sterilization, and what we started with was defining the terms used in sterilization. I believe we have all understood. Is there anyone with a question? I believe everyone has understood. So I'm going to give an assignment which will be delivered next week during our next lesson. The assignment is to explain the classes of sterility indicators and give one example for each. That's it for our class today. Let's meet on our next class next week at the same time and at this same venue. Thank you for attending this class, and have a nice time. Thank you.